Hi, everyone. Good morning. I'd like to introduce Dr. Monica Guzman. Dr. Guzman is an associate professor of pharmacology in medicine at Weill Cornell Medicine. She obtained her PhD in microbiology and her postdoctorate in leukemic cell biology from the University of Kentucky. Dr. Guzman's primary research interest is the identification of novel therapeutic approaches for targeting leukemic stem cells without harming normal hematopoietic stem cells, demonstrating that this is possible in vitro ablation of these cells without harming normal cells by targeting unique molecular features present only in leukemic stem cells. Additionally, Dr. Guzman has received the Irma Hirsch Investigator Award, was listed in the 100 most inspiring Hispanic scientists in America, and received the Faculty Diversity Award. Thank you, Dr. Guzman.
Well, thank you very much for the introduction, and thank you very much for the invitation to tell you a little bit about what is going on in the lab. It's great to be in Miami, even though I'm remotely visiting, but it's good to interact with people, even via Zoom. So today, these are my disclosures that I have to present. So I think by now we all agree that one-size-fits-all approach is not the optimal way to treat patients. As we start identifying mutations and new drugs are coming out, we know that having more tailored therapies is possible. But what we also have to think, even though we want to focus, for example, in elephants, we know that elephants can be different from each other. And for example, in the case of today, I'm going to talk to you about AML. So even though we think about AML patients, we know that each individual patient might have a different type of AML. But even within a same elephant or a same AML patient, there can be heterogeneity that we have to consider when we are thinking about therapeutic approaches. And I hope to convince you that we need to see things that way as well. So in general, the overview for today's presentation will be, I'm going to discuss a little about leukemogenesis, leukemia stem cells, the feasibility of targeting leukemia stem cells, targeting the giving 3 examples. One of them is targeting the immunophenotype, targeting the epichaperome, and I'm going to explain what the epichaperome is, and targeting iron metabolism. So briefly, I think you all are all well familiar with this, and acute myeloid leukemia is, even though it's a rare disease compared with breast cancer and prostate cancer, for example, it has really dismal outcomes for most patients over 55 years old. And this is just, even though it's not updated, the overall survival rate has not changed dramatically over the years since therapeutic approaches have started for this disease. Right now, the 5-year survival went from 26.6 to 29%. And in general, the standard of care for AML has changed somewhat in the past few years, actually, even though the therapy was identical for over 4 decades. However, even though we have newer therapies, patients are still relapsing, and even though they can achieve complete remission. So it's still a disease that we need to think and identify new approaches to target it. So when we think where leukemia stem cells come from, it has been postulated that either a hematopoietic stem cell or a hematopoietic progenitor can acquire one or several oncogenic events. This will result in a leukemia stem cell, and this is a leukemia stem cell that has the same capacity to self-renew, just like a hematopoietic stem cell. But in this case, it will give rise to a leukemic progenitor with limited capacity of self-renewal, and this leukemia progenitor will give rise in turn to the leukemic blasts. By now, we have actually evidence in humans that this can happen, and our group and other groups have shown that there is actually, you can identify pre-leukemic mutations in healthy individuals that have not been diagnosed with the malignancy. And this can be identified even 10 years before the original diagnosis of AML. And this is just an example of this large cohort that was sequenced where we identify several pre-leukemic mutations and chip mutations in AML patients prior to the diagnostic of the disease. However, for today's presentation, it's important not to think about where leukemia stem cells come from, but where leukemia is comprised. And in this case, it's a heterogeneous population of cells that consist of leukemic blasts, leukemic progenitors, and leukemia stem cells. And this has an impact in therapy because when a patient comes to the clinic and receives the standard of care, the majority of the leukemic blasts are going to get eliminated. However, some population of cells, which will include the leukemia stem cells, are left behind. This window is what we know as a window of potentially measurable residual disease, or MRD, in which later on the leukemia stem cells, because it's a capacity to give rise to progeny, will be able to recapitulate disease and give rise to the leukemic relapse. The clinical evidence of the implication of leukemia stem cells in poor outcomes has been shown now for several years, where initially when leukemia stem cells were defined as with the phenotypic markers C34 positive, C38 negative, it was shown that patients that have higher proportions of 34 positive, 38 negatives assessed by flow cytometry would have poor outcomes, and they will have also higher levels of minimal residual disease and led to poor survival. So when this field started over 20 years ago, and I started working on this as a graduate student, these two populations were pretty much identical by phenotype. They were defined as C34 positive, C38 negative, DR negative. The difference was that one gave rise to leukemia and the other one gave rise to whole normal blood. By now, there's several immunophenotypic markers that have been identified and described to be unique to leukemia stem cells and absent in hematopoietic stem cells, or present in hematopoietic stem cells and absent in leukemia stem cells. This has helped the field advance greatly because that allows us to isolate these populations of cells and evaluate their biology. It is important to mention that this list that I'm showing here is not complete by all means. It's just a representation for you to see these differences. There's many more markers that have been defined. But the other thing is important to point out is that these markers are not universal, meaning they are not going to be present in every AML patient to define their leukemia stem cells. There will be some patients that maybe they will express IL-1RAP, and some other patients that will express, for example, CLL1. But in general, the presence of these aberrant markers will indicate that there is a leukemia stem cell in the disease. In addition, it has been also shown that patients that have the present this putative leukemia stem cell at levels higher of 0.1%, these patients are going to have poor outcomes compared with the putative leukemia stem cell negative. Importantly, when this definition of putative leukemia stem cells is combined with standard definitions of MRD, you can see that the stratifications of patients can be improved in the sense that we can separate the ones that are going to do really poorly versus the ones that are likely to do much better. However, having the ability to stratify patients is not sufficient. We need to be able to identify ways to eliminate this population in order to improve outcomes for these patients. I'm just going to also mention that just the immunophenotype is not sufficient to define a leukemia stem cells. The most optimal way to define a leukemia stem cell will be by function. And what this means is performing assays that will tell you the function of this particular cell that we're evaluating. For example, if we take bone marrow cells from a healthy donor and we plate them in methyl cellulose, we will be able to assess normal formation of blood colonies. In contrast, if the sample comes from an AML patient, what we're going to observe is the formation of leukemic colonies. And this will indicate the progenitor cells. And the gold standard assay to assess stem cells would be to the transplantation in the immunodeficient animals. In the case of normal hematopoietic cells, when they are transplanted into an immunodeficient animal, the animal will have human blood cells. And when the cells are coming from a leukemic patient, the animal will develop leukemia. And by transplanting into secondary animals or tertiary animals, we're able to show that these cells have the capacity to self-renew. And by being able to initiate disease again and again and again. So what my laboratory has focused now for several years is to determine differences between normal hematopoietic stem cells and leukemia stem cells with the purpose of eliminating the malignant cell without harming the normal hematopoietic cell. And that's where I have this example where we have the Spider-Man that was evil versus the good one, and that's kind of to represent that point. So there are many different approaches that have been described to date to target specifically leukemia stem cells without harming the normal counterparts. Here is a list of many of them. For example, the unique immunophenotype, iron metabolism, mitochondrial function, signaling networks, survival signals, the epigenetic machinery, the leukemia stem cell niche interactions, oxidative metabolism, and the epichaperone. In red, I have highlighted the 3 examples that I'm going to discuss today. So first, the immunophenotype. And the immunophenotype is one of the most common ways to try to target the leukemia stem cell, since it's one of the first ways that these populations, or difference between these 2 populations, were identified. And one of these first differences was the expression of CD123 in leukemia stem cells. So CD123 is the IL-3 receptor alpha chain and is expressed in several myeloid cells. These will include plasmacytoid dendritic cells, basophils, monocytes, eosinophils, dendritic cells, in addition of myeloid progenitors. It is important to mention that the expression of hematopoietic stem cells is very, very low to none. And the other important feature to this is that CD123 is expressed in the leukemic blasts, in the progenitors, and the leukemia stem cells. So it makes it an ideal target because we can eliminate, in theory, the entire population of cells. And this is just an example for you to see that even though CD123 is expressed on normal cells, shown here in blue, there is still a therapeutic window when we assess the expression of CD123 in AML patients. The approach we were using to do this is the use of chimeric antigen receptor T cells or CAR T cells. As you know, these are engineered receptor to target surface molecules, in this case CD123. And the important thing of this is that their engagement to the target cell is independent of antigen presentation. And this is done by using a single chain variable fragment that is designed to bind to the particular target. So the most typical approach that we heard about CAR T cells is the autologous CAR T cell process, in which the T cells are taken from the cancer patient, they are activated, they are transduced, expanded, and then put back into the patient. The process I'm going to discuss today are allogeneic CAR T cell or universal CAR T cells. These come from a healthy donor where the T cells are purified and isolated, then they are transduced, engineered, and amplified. So we can freeze several vials. So this will be an off-the-shelf product, and then they can be used in several AML patients as they walk in the clinic. So in order to achieve this, these CAR T cells have to have very particular features. And the features of the CAR T cells we have used is one, they can detect CD123. They are a second generation CAR. And an important feature is that they have the TCR alpha beta knockout using TALENT technology. This will decrease the risk for graft versus host disease in the recipients. In addition, it has a safety feature which consists on having the expression of the RQR8 gene. This is a construct that allows or renders the cells sensitive to treatment with rituximab. Or the products or the CAR T cells I'm going to discuss today, they were provided and generated by Selectis as part of a research alliance we had with this company. So the very first thing we did was to use these UCART123 cells in vitro. So we did liquid cultures using different primary AML patient samples. And you can see here that when they were treated with different effector to target ratios, we were able to kill the majority of the AML cells using effector to target ratios as low as 0.5 to 1, in contrast with the TCR alpha beta knockout cells that have no CAR. So we also did colony forming assays, and you can see that when the cells were co-culture 1-to-1 ratios, the CAR T cells eliminated the progenitor cells shown here. We did the same assays in normal hematopoietic cells, in this case using cord blood cells shown in here, and the liquid culture show no harm on hematopoietic populations. Of course, since we are concerned about what would happen to the myeloid lineage, because CD123 is expressed in hematopoietic progenitors, myeloid progenitors, we did colony forming assays. And as you can see, at the higher effector to target ratios, as expected, we see some decrease in myeloid colony formation. However, at effectors to target ratio of 0.5 to 1, that we know is effective to kill AML cells, we saw a minimum to no effect on this myeloid colony formation. So we then proceed to test this in vivo using patient-derived xenotransplants, in which we injected primary AML cells into sublethally irradiated mice. Once we established the xenograft, we treated the animal either with UCART123 cells, the control T cells, or cytarabine, as is one of the drugs used in the clinic. We started monitoring the animals at day 2, and then we continue evaluating continuously and determine survival. As you can see here, in this axis, we're showing the % human cells in the peripheral blood of the animals. And you can see that after starting RSC treatment, the % of leukemia cells in the peripheral blood decrease as expected, as observed also in patients. However, 1 week later, these animals started to relapse, develop as much disease as the controls, and you can see eventually all died. And the benefit of survival is minimum, again, as seen in the clinic. When we use the TCR alpha beta knockout cells, there is no benefit. The animals die just like the control saline. When we use the CAR T cells, we saw a decrease in the level of disease in the peripheral blood observed here, with doses of 1 million and 2.5 million. When we look at the overall survival, these animals have a significant improvement on overall survival. It's important to point out here that when these animals treated with 2.5 million cells and they started to die, these animals were not dying of AML. They were dying of old age, because by the time we started the experiment, the animals and the time we were observing, the animals were getting quite old. So we decided to terminate the experiment at day 221 and evaluate what was happening in these animals. So as we were looking at this, we also wondered what happens in this window. Even though we cannot detect exactly the disease in the peripheral blood, as you can see by flow cytometry, since the number of events or the % of events observed are very little, in this case 0.14, for example, very few dots, this is not going to allow us, or was not allowing us to determine if these few events were coming from the leukemia or from the CAR T cells, which are also of human origin. So this PDX in particular had an NPM1 mutation. So we were able to design a digital PCR assay that will allow us to have more sensitivity to, one, monitor the residual disease, and two, also monitor the CAR T persistence. So in addition, this assay will allow us to measure the total human ABL, and also allows us to measure the mouse ABL as a control to let us know that we took a good blood sample. So what we observed was interesting, but because this is what we observed with the 1 million dose animals. We found that when we are able to detect the CAR T cells, here you can see the levels of transcripts are around 100. But if this transcript decrease of the CAR T cells over time, the mutation, the mutant cells start to come back up and the animal relapses. And this was confirmed when we sacrificed the animal. In contrast, when the CAR T cells were not, were actually persisting, shown in here, if you can see the levels of overall transcripts are very similar. The axis is different, but the total amount of transcripts is very similar. But the transcripts remain and eventually start to increase. You can see there is a decrease that is consistent with this increase of CAR T cells of the tumor cells, indicating that the CAR T cells were able to continue eliminating the NPM1 mutant cells. And this animal was disease-free by the time we sacrificed it at day 221. So this was very nice because it was showing us that the CAR T cells are very effective at eliminating leukemia, but we still had concerns about what would happen to normal hematopoietic cells. So we performed several humanized models, but the data was very variable from humanized mouse to humanized mouse. So we set up a competitive xenograft where we transplanted bone marrow cells and AML cells. So we transplanted normal bone marrow CD34 positive cells as well as primary AML cells, with a difference, with haplotype difference, in this case HLA-A2, so we are able to determine which ones are leukemic, which ones are normal. Once we confirmed that the animals were chimeric, we started treatment with the CAR T cells or the control T cells. As you can see here, by flow cytometry, we were able to show that the CAR T cells selectively eliminated the AML cells without affecting the normal populations. When we looked more closely at the populations that survived, we did see a decrease, as expected, in myeloid cells, around a 60% decrease on myeloid cells. However, the CD34 positive progenitor cells were not affected. We also confirmed by digital PCR that the NPM1 mutant cells were going away. So these animals were achieving molecular remission in the assessed in the peripheral blood. And at day 36 in the bone marrow, we also observed that there were no leukemia cells in the relative to normal in the bone marrow. Again, showing that these CAR T cells can selectively target AML cells without sparing normal cells, without harming normal cells. So the summary of this first part is that UCART123 cells is an off-the-shelf product of allogeneic engineered CAR T cells and can effectively eliminate AML cells in vitro and in vivo. CD123 was significantly able to improve the overall survival of PDX in mice. And right now we are in a phase 1 clinical trial for UCART123 in humans. And just as an example, we were able to apply the same assays developed, for example, the digital PCR in humans. And this particular patient had an NPM1 mutation. And you can see here, we are able to assess, at day 14, we're able to detect the CAR T cells in this patient. And we're able to observe also the decrease of NPM1 mutant cells in peripheral blood and bone marrow. And again, these studies are currently ongoing. And this is just briefly to point out that not every CAR T cell targeting leukemia stem cell immunophenotypic marker works the same way. Here we also we're trying to target CLL1. And you can see here, even though in vitro, these CAR T cells are very effective at eliminating CLL1 positive cells, we did not see such a significant, as we did with UCAR123, improvement in overall survival in mice, as shown here. Even though it's significant here, the difference is not as impressive as with UCAR123. Again, suggesting that each marker might have a different activity and a different way of action. So the next example I'm going to talk about is the epichaperone. And this is a work in collaboration with Gabriela Chiosis and Memorial Sloan Kettering. So how to define the epichaperone? The best way to define it is using the following analogy. And I guess this is not going to be as interactive because we're in Zoom, but if I were to ask you, how do you feel when you misplace your smartphone? I guess most of us are going to be here. We feel like our world is falling apart. And sometimes the main reason is because all our networking capacity sometimes is linked into our smartphone. Meaning probably many of us don't remember many phone numbers, addresses, how to get to a place. We have access to that through our cell phone. So if we forget it, it's likely that maybe we're not going to be able to find our friend. We're not going to be able to find a particular home. But if we use our cell phone, our smartphone just as a cell phone to just make phone calls, for example, you will feel relief. Why? Because even if you lose it, you will find alternative ways to network. You might be able to get to that place, to get to your friend without the need of using your smartphone. So the epichaperome is something very similar that is found in some cancer cells, in which they are very hyper-connected networks of proteins that nucleate with HSP90 and HSP70 machinery. So you can imagine if you take out, for example, HSP90 or HSP70, the whole network will collapse and the cell won't be able to survive because everything is tightly connected. In contrast, in some other cancer cell, these networks are much smaller. They also nucleate in 90 and 70, but if you disrupt 90 or 70, the cell will find alternative signaling pathways to survive, and it will not be affected by the disruption of these machineries. So as we were defining this, we also developed chemical tools that will help us assess or evaluate the levels of this epichaperone in cancer cells. And as you can see here, we were able, using this, we call it PU-FITC. We use this PU-FITC probe that allows us to identify, it will bind to the epichaperone. It binds specifically to HSP90 when it's part of this epichaperone. And we can measure by flow cytometry on cells that will have HSP90, but is not in the conformation of the epichaperone. PU-FITC is not going to bind. And these cells are going to be not FITC positive when we evaluate by flow cytometry. What is also important is when we use the epichaperone inhibitor, which is called PU-H71, cells that we know have high levels of epichaperone are going to be killed by PU-H71. In contrast, cells that are epichaperone negative, they are not going to be eliminated by PU-H71. We have shown now across tumor types, but specifically here, the example is AML, that there is a correlation between the epichaperone abundance and the sensitivity to PU-H71. Importantly, in PDX animals, when we use AML cells that have high levels of epichaperone, these cells, these PDXs, the AML cells in these PDXs are going to be sensitive to PU-H71 treatment shown in here. When they have low levels of epichaperone, these cells are not going to be as responsive to epichaperone treatment, inhibitory treatment. It's also important to point out that another thing that we found is that AML that have high levels of epichaperone have also hyperactivated signaling networks shown in here. And cells with low epichaperone abundance do not have this hyperactivated signaling. Importantly, when we did secondary transplants using the cells from these PDXs that were treated with PU-H71, we found that cells that have high epichaperone abundance failed to recapitulate disease in secondary animals. And in contrast, cells with low epichaperone abundance, even though they respond a little bit to PU-H71 treatment, when transplanted into secondary mice, actually led to higher levels of disease in these animals, suggesting that we were probably eliminating more mature cells and enriching for leukemia stem cells or cells with initiating capacity. But we had all this data and we were wondering if we could use this test or this assay in humans that would allow us to tailor therapies for AML patients and identify those patients that will be more likely to respond to this therapy. So this is a huge effort that was done across many different disciplines. And I'm going to tell you this particular case report, but this is the list of all the contributors on this particular case. So this was a 61-year-old woman that was diagnosed with accelerated phase of myeloproliferative neoplasm. This happened in 2013. She was negative for all the typical mutations or rearrangements expected for MPNs, and she was also negative for JAK2 mutation. She underwent to match unrelated donor allogeneic stem cell transplantation in 2004 because her disease was progressing. In 2016, she relapsed from her transplant and she started treatment with hydroxyurea. She had an atypical presentation of GVHD in one of her toes, and that was resolved with ruxolitinib. In 2017, she started progressing to AML with fibrosis, and she started to develop splenomegaly, weight loss. She continued to worsen, and she was not responding to any of the therapy she was given. So as part of what we have here at Weill Cornell is we have the Institute of Precision Medicine, in which cases like this, where they are unique, they are different from what is expected, they are sent to them to be sequenced. So these patient cells were sequenced, and what they found is that she had a novel gene fusion between PML and SYK. And because the urgent need for her to receive therapy, we started to try to determine what this fusion protein might be affecting in order to identify new therapy for her. So first of all, we validated that the PML-SYK translocation was present in her cells, and we sorted lymphocytes, blasts, and granulocytes. And here you can see that the granulocytes and blasts are positive for PML-SYK. And the reason that lymphocytes are negative is because the lymphocytes come from her donor. So the only thing she has left from her bone marrow donor are the lymphocytes. Because at the time, we didn't know what would be affected, a postdoc in the lab, she started looking at the signaling pathways, especially because SYK was involved, but she pretty much tested every phospho antibody we had in the lab for flow cytometry use. And she found that every signaling network was on, which led us to think that maybe because she has hyperactivated signaling, she might also have high epichaperone levels. So we tested out our PITC probe and found that the blasts and the granulocytes were epichaperone positive. We treated these cells in vitro with the epichaperone inhibitor, PU-H71, and found that the cells were sensitive to treatment, almost as the control cells MB411s. And you can see here, they respond very well. So this led Dr. Gail Robos here to start a single patient IND for this particular patient using PU-H71. So as part of the treatment, we evaluated the patient, we followed the patient's CBCs, we evaluated signaling networks and epichaperone abundance. So initially the treatment schedule was exactly the same of what was used for breast cancer, which is 2 times for 2 weeks, 2 times a week for 2 weeks, then a week rest, and then start again. So you can see it here reflected that when the patient started treatment, her white blood cells came back to almost normal range, her blasts started decreasing, her spleen size also started decreasing. But when we came to that holiday, cells started to come back up again. So that led Dr. Robos change the schedule of this patient. So she was given 2 times a week continuously until she was able to achieve complete remission. Here, I'm showing you that, again, as she started treatment, the PU-FITC binding decreased as expected. So this shows us also that we are achieving target engagement. And the signaling networks start also decreasing after she started treatment. Importantly, when we look at the stem cell populations, we see that the phosphorylation of SYK, shown here as an example, is starting to decrease over time as she's receiving treatment. Again, suggesting we're also affecting this population of cells. When we look at the bone marrow at comparing baseline with day 43, after treatment, this is when she achieved remission, you can see we're starting to see the formation of normal red blood cell colonies, suggesting that things are improving in her bone marrow. And this is just to show you a timeline. So the patient is still alive, she is still in remission, and she is still coming for her therapy every 2 weeks. And you can see here that she achieved the complete remission, and after she achieved the complete remission, her treatments start to decrease on periods of time. We found that if we wait more than 4 weeks, the blasts start to come up a little bit in the peripheral blood. So she comes for treatment once every 2 weeks. Another interesting thing we found is that her monocytes, because that's the thing we see changing the most right now that the patient is in remission, her monocytes are also positive for the epichaperone compared to normal monocytes. And over time, we saw a change on the proportions of classical, intermediate, and not classical monocytes. And eventually, over time, the proportions were closer to what is expected in normal individuals. This suggested that maybe since she received a transplant and the lymphocytes are her donor, that maybe PU-H71 might be also having an effect on the lymphocytes that are helping to maintain her leukemia low. So this inspired these experiments that are still ongoing, in which we use the OVA antigen challenge model, in which we observe that PU-H71 can significantly improve the interferon release of this when it's combined with the OVA antigen. And it's shown here, you can see it here. This is OVA by itself, OVA with PU-H71, and this is relative to the positive control. And this change is significant, and this has been repeated several times. So right now we're trying to evaluate the potential impact also of PU-H71 in T cell function in the context of planning a clinical trial for patients like this particular patient I have discussed today. So the summary of this part is that using this novel flow cytometry assay, the PU-FITC binding assay to evaluate epichaperone levels, we were able to identify or predict the response of this patient that needed urgent treatment. This patient had relapsed even after allogeneic bone marrow cell transplantation, and right now she is still in remission. It's been over 2 years. So And lastly, the last example I want to discuss, it's a little bit different because in this case, we are trying to take advantage of the lack of expression of a protein or a transporter. This is a collaboration with Jan Grimm, also Memorial Sloan Kettering, and this is ferroportin. So ferroportin is the only known iron exporter in human cells, and it plays a very critical role in exporting iron outside the cells. What we found is that the expression of ferroportin was decreased in leukemia cell lines and in primary AML samples. And this was confirmed by flow cytometry. We saw that the majority of the cell line had very low levels of ferroportin, as well as primary AML samples. As you can see here, as we go in the more primitive populations, the levels of ferroportin are very low. So that led us to hypothesize that maybe if we had a way to put more iron inside these leukemia cells that have low ferroportin levels, this iron won't be able to get outside the cell and will likely lead to oxidative stress, and these cells will die. In contrast, normal cells or cells with high or normal ferroportin will be able to export out the iron and still survive. So what we use is ferraheme or ferroxytomol, which is indicated for treatment of iron deficiency anemias in adult patients. They can be used with intolerance to oral iron or people with chronic kidney disease. So ferraheme is composed of supermagnetic iron oxide nanoparticles, and they have a semi-synthetic carbohydrate coating. And what this makes it special is that eventually we can also load them with other drugs. So, and also the iron ferraheme is easily utilized by the body and is incorporated in iron stores. So the first thing we did is we tested these nanoparticles in vitro using cells with low ferroportin and cells with high ferroportin. And you can see here, there was a significant decrease in the survival of cells that have low levels of ferroportin when treated with ferraheme. So we went to an in vivo model. In this case, we used this model of acute leukemia or blast crisis CML. And we treated with saline, cytarabine, or 3 milligrams per kilogram of ferraheme or 6 milligrams per kilogram of ferraheme 2 times a week. And we found that there was a significant decrease when the animals were treated with 6 milligrams per kilogram of ferraheme, looking at the peripheral blood and the bone marrow, and also the spleen, although it's not shown here. We also observed a significant improvement of overall survival on these mice after treatment with ferraheme. After this animal model, we also wanted to test PDXs. And in the PDXs, we transplanted either cells, leukemic cells from patients with low levels of ferroportin, high levels of ferroportin, or normal cord blood cells as a control. Once the PDX was established, we started treatment of these animals for 4 weeks, either with saline or ferraheme 6 milligrams per kilogram. And then after that, we evaluated the bone marrow for tumor burden. And as you can see here, leukemias that have low levels of ferroportin responded to ferraheme treatment, shown in here. There was a significant decrease on the overall leukemia burden in the bone marrow of these cells. In contrast, the animals with high ferroportin had no significant difference in the overall tumor burden after treatment. And importantly, animals transplanted with normal cord blood cells had no effect or no difference in the total amount of cells in the bone marrow. So just to summarize this part, I discussed that by taking advantage of the decreased expression of ferroportin in AML cells, we were able to eliminate leukemia cells using ferraheme. Importantly, ferraheme particles can be loaded with other compounds to prove effectiveness of therapy, and that's something we're testing right now in hopes to see the feasibility of a clinical trial. And I just want to show you pictures of the people in the lab. And the reason they're holding a timer is because we had the opportunity to meet the inventor of the digital timer. And we wanted to show him that we actually use these timers, probably not for long because now everybody will be using the cell phones. But I thought he found it quite interesting to see everybody so excited about this invention. So thank you very much for your attention. And oops, I want to take this out. And I'm happy to take any questions.
Thank you, Dr. Guzman, for that very excellent and inspiring translational talk that was very clearly presented and very interesting. Can we go back to your epichaperone patient who seems to be getting this chimeric treatment, the PU-H71?
Mm-hmm.
How were there any side effects? You didn't mention any, and is this something that she's going to need to be on for life? Because it seems as though it's like weeds; you sort of just mow them down and then they come back up again. Is that the concept?
Yeah, so I'm really happy you asked this. So the first part, the side effects were minimum. She had some diarrhea, not severe, and some aches and pains in her joints, and we think it's probably because we're also having an effect on her T cells, and she had some levels of GVHD. So that's one. And so that's great because she pretty much felt much better. She was very happy to not having that splenomegaly. That was the very first thing that was obvious that happened. But if we go back to the case, it started as an MPN and then transformed to AML. So what we think is happening is that that we were able to get rid of the AML, but not of the MPN. And that PML-SYK still present. So when we sequence and we evaluate her cells, PML-SYK still there. We don't know how it started or what is the causative effect, but this is turning more like a CML, like the CML story, viscerable. So in order to keep this initiating cell that is bearing PML-SYK, that is not dependent in this case of in the epichaperone for survival, if we stop the therapy, this cell starts to come back, and what we're seeing is what is coming back is her MPN and not her AML. So yes, she probably will have to be in this therapy. We're trying to see if we can find other options to help her, but in the meantime, she will require to continue PU-H71 treatment.
Thank you.
And we keep monitoring her to make sure it still will work. That's the whole point, to continue the epichaperone assays, just to make sure she doesn't generate resistance and all of this ahead of time.
Thank you. Dr. Sekris, do you have any comments?
I do. First of all, I want to thank you, Dr. Guzman, for speaking to us today. I want to join the people who voted you one of the most 100 inspiring scientists around. You always present such interesting research and so clearly. I want to build a little bit on what Dr. Weiss just asked about how we target patients who have leukemia cells with abnormalities. Do you think we'll ever reach a point where we can have monotherapy that's tailored to a patient's leukemia, or is leukemia just too clever with too many escape mechanisms, and maybe we're not targeting the right protein expression in the hierarchy of what gets expressed as leukemia cells arise?
So when you say that those are my nightmares every day, pretty much, because the more we know, the more we sequence, and I think I showed you the example about the pre-leukemia mutations just as a background. And we know that these mutations can happen even 10 years before the disease is diagnosed. And to me, that was when we first found it, it was like, wow, I thought that, we didn't know, we couldn't predict leukemia, it just happens. And it turns out that it's lurking there, it's starting, you have pre-leukemic events, you have pre-leukemic hematopoietic cells. So to answer your question, until we don't know what is the source, and I think maybe eventually will happen, until we don't know what is the true root and the initiating event, it's going to be very difficult to eliminate the disease. But I think in order to minimize the risk of getting it more aggressive, we need to know how to hit it hard and hit it hard from the beginning. And I think the key might be to have therapies that can work during MRD, so you can hopefully eliminate that residual clone. And that's the challenge, because the challenge is how we measure. That's what was switched from minimal to measurable, because it's what we can measure and we can see. But I think that will be the key. And sadly, to me, it has always been like that game of whack-a-mole. You press one and it's like, I got it. And then the other one comes out. So we need a bigger hammer. And until we don't find that, we won't know what is the true answer. And I know we all got very excited with venetoclax azacitidine combo, but we're seeing relapses again, and they're very different. So there is a lot to learn, I guess.
Oh, absolutely. And I'll just for others who may not be aware, the study that Dr. Guzman's referring to that was led by Dr. Desai was actually from the Nurses' Health Study. And I think it took Dr. Desai about as many years to finally get permission to publish as it did for those genetic abnormalities to evolve to leukemia for people on the study. But it showed that those who have certain discrete abnormalities are basically guaranteed to get leukemia a decade or more later. It's very frightening statistics, and that's what we're dealing with as we establish our own CHIP clinic and clonal hematopoiesis clinics at a variety of centers.
Yeah, thank you.
Great. Well, I want to thank everyone for coming to Grand Rounds today. Don't forget to sign in and fill out the questionnaire and claim your MOC credit. If anybody has any other questions, feel free to write them in. We'll forward them to Dr. Guzman. I haven't been following the chat closely, but anybody wants to remain on, please do so. Thank you, Dr. Guzman. Enjoy the 81-degree weather in New York. It won't last.
Thank you. Very nice to see you all. Bye.
Bye.
Thank you, Dr. Guzman.
Thank you.